Okay, I hope you all had enough time to finish the recap quiz. Let's get started with the lecture now. So, in today's lecture, we are going to be covering cell tracking. So, specifically, we will use this plugin called Trackmate. I'll also show you a Fiji demo. First, I'll tell you about the important features in Trackmate, and then I'll show you how to do it on Fiji. So, Trackmate has different steps. Um, The first step is spot detection. So if you give an image, first of all, or if you give a series of images for tracking, first of all, it has to detect where the objects are present. That is just segmentation. And then the second step is spot filtering, meaning that if the algorithm has detected maybe 20 spots, but not all of them are true objects, maybe it has done over segmentation, then there is an option to remove spots based on size, sphericity, visibility, other physical features, etc. So once the spots are or objects are segmented and corrected, then the next step is tracking. So there are different tracking algorithms like linear assignment problems, nearest neighbor tracker, etc., which also I will briefly talk about in the slides. And after tracking is done, there is also an option to filter the tracks. So not all the tracks that the TrackMate detects may be correct. It might sometimes do wrong tracking. So there is also an option to remove such tracks by setting threshold parameters. And once the tracking is done, you can also export the outputs in different formats. And there is an also finally an option to um, change how the tracking output looks. For example, you can color give colors to each different object based on their area or shape index, like a heat map sort of. And you can also color the different tracks based on the speed of the tracks or speed of the objects, etc. So these are the um, this is the basic workflow of TrackMate. So the first step here is spot detection. There are different detection algorithms, like it uses uh, a logarithm of Gaussian approach, DOG approach, etc. So in these methods, the segmentation is done on TrackMate itself, and it works when, for example, if you have images that have a clear distinction between background and the object, then it works well. But in biological images, it may not always work well. So there is an option to directly give segmented images as input to cellpose, and from the segmented label images, it will then detect the spot. So this is the method that we will be using for our kind of microscopy biological data. So this may not work for microscopy biological data. So that's why we are um, directly feeding the segmented images to TrackMate. So one more option here. This is one option to first segment the cells on TrackMate and then give it as input. There's also another version of TrackMate which includes cellpose. So you can use cellpose within TrackMate. That is another option. You can also that that can also be done. But for in this lecture, we will be mostly covering this method. So we will be using the label detector in TrackMate because we are going to give our input is going to be segmented label images. that we get from cellpose. So this is the first step, which is spot detection, which we have now covered. And then you can also filter the spots. Uh, just a minute, let me just. Yeah. So after detection, you can do spot filtering. So what um, Trackmate does is first it detects the spots and then it calculates features like physical morphological features of the spots. So this can be things like area, sphericity, um, shape index, um, etc. So once it has, and this it does this for all the images that we give. So because for tracking, we generally give 2 or more images. Um, and similarly, first it detects the spots also for all the time-lapse images. And once this is done, you have an option to filter the spots based on the physical features that Trackmate has calculated. So, for example, if say you had used cellpose, but cellpose has had done maybe over segmentation. So maybe your microscopy image had some cellular debris and it had also detected the debris. So if you have cells which are of the size 20 microns, but cellpose has also detected debris which are smaller particles, maybe 5 microns, then you can filter this by area on Trackmate. So you can have a size cutoff basically and ask Trackmate to remove spots that are maybe if this all the cells are 20 microns and above, then maybe and the debris is about 15 microns, then you can set a cutoff of something like 15 to 20 microns to remove the wrong segmentation. So this is helpful when you do cellpose with high flow threshold and low cell probability threshold. Sorry, this is cellpose. For example, if there may be some images for which for cellpose to detect all the cells, you may need to use a high flow threshold and a low cell probability threshold. But when you do this, sometimes cellpose also detects the smaller debris and over segments the image. So this can be then fixed by using Trackmate. So in the previous class, we used a plugin called Labels to ROIs, which can do this. But another way to get the physical features of each of the cells is also by using this plugin called Trackmate. So this is the second step, which is spot filtering. So after filtering, you have tracking. So tracking, one of the most, I'll just discuss two of the very commonly used tracking algorithms on Trackmate. There are others as well. So the first one, maybe I'll talk about the others when I show you the Trackmate demo. So the first tracking algorithm is the simple lab tracker or a linear assignment problem. So here, so basically tracking means if you have objects or segmented objects in two different images, then the algorithm has to identify that this object in the first image is the same as this object in the second image. So this can be done by matching how far the two objects are, comparing the sizes of the uh, particles, comparing the intensity of the particles, etc. And if two objects are closer in size, then they have a lesser cost. Um, and similarly, if two objects have similar intensities, then also they have a lesser associated cost. So it's more likely that those two objects will be considered as the same by the tracking algorithm. And another simple approach is called the nearest neighbor tracker. So this is a very simple algorithm which just compares the segmented objects in two different images. And it says that the object that is closest in the second time point to the object in the first time point is considered to be the same object. Obviously, this may not work well when particles are moving quite fast and they exchange neighbors and so on. So, when you have a jammed system where the particles don't move much, then maybe this method will work. So, this method works only in very specific cases. So, again, depending on the kind of data set that you have, you will have to pick which tracking algorithm to use. So, it's good to know the basics of all of the tracking algorithms. So once tracking is done, you can also do a track filtering. This is similar to spot filtering. So where after detecting the tracks, cellpose then will then calculate different features of the tracks, um, such as for how long the track is visible, or the speed of the track, the directionality of the track, and so on. And then based on these features, you can then set cutoffs based on these features to remove tracks that are not done correctly. And once track filtering is also done, then basically you just have to export the output. So Trackmate has different forms of outputs, which will be more clear later on. So the main Trackmate output is a .xml file, which will have all the information about so it's like a file with all the metadata or the settings that you used for all the steps in Trackmate. So the advantage of so this file will be quite heavy because it has a lot of information. But the advantage of having or saving this file after you do the analysis is that the next time you can also import it into Trackmate directly if you want to later check what parameters you used or if you want to change something in the analysis, um, and so on. So this is the main output. And apart from that, you also get CSV file outputs called spots.csv and tracks.csv. So these are like Excel sheets which have information about each spot over time. So it will be able to say that it will have columns like the spot number, um, the time point or the time, depending on uh, whether you have given the conversion factor or not, and then the x coordinate of the spot at that time point, and the y coordinate of the spot at that time point, and other features like what was the area of the spot at that time point, um, uh, many other parameters. These are just some important examples. For example, if there is cell number 1, which is at maybe at the first time point, it is at 0, comma 0, and it has an area of maybe 10 units. This is a very rough example, and a shape index of about 4. But in the second time point, the cell has maybe changed its shape a little bit. It is undergoing, it is in the process of dividing, so its cell area increases. So then you will see that in the second time point, the same cell, sorry, it's the same cell, spot number or cell number in the context of biological data. So the same cell in the second time point has moved a little bit. Maybe it has moved to 1, comma 2, and its area has also increased to maybe 15 units. And because the cell is elongating, its shape index will also increase. Maybe it has now become 4.2. Um, so yeah, I'm not sure if everybody would know what shape index is. So I'll also roughly explain that. So shape index is somewhat similar to aspect ratio. So it's a measure of how elongated an object is, and it is calculated. So it's a simple, it's a dimensionless parameter, and it's calculated as perimeter divided by square root of area. So and this is commonly used in biophysics to describe the shape of cells. Uh, okay, maybe I'll edit this later. Yeah, so that was quickly about. So this is roughly how the spots.csv and this is actually the spots.csv file. This is how it looks. Um, Apart from that, another format to export it is the is something called Motility Lab spreadsheet, which is uh, a very specific format. Again, it is also an Excel sheet with many columns like the above. So these are the numerical outputs. Um, Apart from this, you can also export the Trackmate as a visual output. So basically, it will have all of the cells or the raw image and overlay the cell tracks on top of it. So this is useful when you want to check if the tracking has worked well, or you can also use it to put in your presentations or as supplementary videos in your manuscripts and so on. And so when you overlay the Trackmate on top of the raw image, then it will also give you the intensity of the spot over time. So maybe you have cells that are tagged for, say, actin, and so because the cell is dividing, actin levels, or it could be any protein levels are increasing over time. Maybe initially it was at uh, the average. This is actually the mean intensity. The mean intensity was 200 within that ROI, but later on in the next time point, it has increased to 300. So using this, you'll be able to see how the cells are moving and how they change shape as they are moving. How do protein levels change in the same cell over time and so on. So, okay, my screen is still being shared. So this is an overview of Trackmate. So now let's go to the demo on Fiji. So first of all, you will have, it's a plugin on Fiji. So you will have to install the plugin. Maybe for this, I'll share my entire screen so you can see everything. Yeah. So you can just Google Trackmate. And so this page on Fiji page on Trackmate will have information about how to install it. Um, so and they also have different tutorials and manuals and so on. So this is a very comprehensive document. So this lecture will just be about the basics of Trackmate, but there's a lot more to it. You can also script on your own to make different new features on Trackmate and so on. So you can script it, you can also extend it and so on. So this is how you can install Trackmate. So it's present in the update sites. So you can go to Fiji and check if Trackmate is already installed as a plugin. So here, if you go to tracking, I already have Trackmate installed. So before you open Trackmate, first you have to open the image. So let's see if I have any images for tracking. So these are the images that we had segmented in the previous class. This is T29 S10. Okay, so I think we will need different. So none of these images are time lapse images, whatever we had tracked in the segmented in the previous class. So let me see if I have already put these images for tracking on my drive folder already. Yeah, I think I'll have to download this folder. Um, no, this is not the folder. So let me just download this folder. Um, or I'll just download these two images. So the first step is to segment this on cellpose. So let's first see how the image looks like. Mm, yeah, so this is the folder with the image. Let me close the other stuff. So this is a time lapse image of with two time points. And so these are cells which have very close cell-cell contacts. Um, let's see how they are moving. Yeah, so you can see that they have moved a bit. So based on this image, it looks like the particles have not really moved around a lot. So we can also try different tracking algorithms to see which one works best for this. But first we'll have to segment it. So I'm segmenting the images on the cellpose GUI. But if you have, so these are just two images. So I'm going to segment it on the cellpose GUI. But if you have a time lapse series with maybe hundreds of images, you have done a very long term imaging, then you'll have to segment it using a code because you can't sit and segment everything manually. I mean, this is um, still better than older times where it was not possible to segment these images. So you had to manually open the image and draw the outlines for each image. Now we have come a long way from there, fortunately. Yeah. So on my Mac, I have not installed the Cellpose version with GPU. So I'm first calibrating the diameter. So those of you who have already installed Trackmate, you can also do this along with me. And let's first try with the Cyto 3 model, which would, so you can see that at the cell borders, you see a uh, whitish intensity. That is because these images have been taken in a phase contrast microscope. So the segmentation is quite good, except that it has missed some cells. Most of the cells are detected. It has missed some of the cells here. So let's try to increase flow threshold a bit and now see if it is able to segment more cells. So now it has detected 483 ROIs, but still, okay, it's still segmenting, sorry. Yeah, even now it has detected the same number of cells, not much of a difference. So let's try to decrease the cell probe threshold. So actually my laptop, I have the Cyto 3 model, but if you install Cellpose now, there is a more updated version called Cellpose SAM or segment anything model. Um, so if you have that model, you can run it with that. So now it has detected slightly more. So for example, this was not detected before. So yeah, now it has detected 491 ROIs. And I don't really see any um, see much of wrong segmentation happening. But still it has missed a few cells. So some of the cells that it has missed seem to be a bit out of focus. But there are cells here and here which are in focus, but it has still missed it. So let's try to, and since it has not started over segmenting yet, let's try decreasing the cell prop threshold a bit more. So now it has, earlier it was 491 ROAs, now it is 493. But as you can see here, it has made a mistake. So when you decrease the cell prop threshold, it is starting to make mistakes. Um, yeah, and it has still not detected these two cells. So I think minus 0.4 was working better, although it missed a few cells. So let's just stick to this. Flow threshold of zero 0.8 and cell probe threshold of minus 0.4. So even here you can see that it has made a mistake here. So it has detected some out of focus spot as a cell. So this we can remove when we do the analysis on Trackmate. And similarly here also, you can see that both the mistakes it has made, it has detected small objects that are not cells as cells. So I'll save this output. So now this is saved here. Now let's also segment this with the same parameters. I'm not calibrating it again because the two images are similar. So I'm just running it directly with the same parameters that we had optimized for the previous time point. Yeah, so here it has detected slightly more cells. Um, so you can see that there is one cell division over here, um, and one cell division here, and one cell division here. So it makes sense that the number of detected cells should actually, there's also one more cell division here. So yeah, so it makes sense that the number of detected cells has increased, and you can also see that this cell is probably about to divide. Um, yeah, okay. But also, yeah, and even in this image, you can see that this part of the image has a better focus compared to this part where the focus is the cells are slightly out of focus, but so cellpose has missed some cells here, but not too bad actually. And even in this corner, the focus is slightly different. So it has also missed some cells in this region, but it's okay. So the better, if the cells were also in better focus here, then cellpose would have detected it also. So when you are acquiring images, when you're doing an experiment and you know that you will be doing cell segmentation for your analysis, just try to um, make sure that the images are in focus when you acquire them, especially when you're doing a time lapse imaging, because sometimes over time it's possible that the cells can go out of focus. So you'll have to keep checking the imaging to see if the cells are still in focus. If not, you'll have to readjust the focus and so on. Okay, so now these two are saved. So these are the mask images. So we see that whatever cells it has probably missed in the segmentation has missed in both the time points. And before doing Trackmate, you can also calibrate the image. So so it has detected it as a Z-stack image. So I'm converting it into a time series image. And this was taken in a 20x objective. And maybe the frame interval was, they were 10 minutes apart. So after you set these parameters, then you can go to Trackmate. So now it has taken the um, pixel to micron conversion that we just set. So then the final um, outputs of Trackmate will be in these units. So here you also see which paper to cite if you are using Trackmate. So we are using, so there are different detectors like I was telling in the lecture. And when you click on it, you'll be able to see a bit about how the algorithm works. So if you're not sure which detector to use, you can also read about each algorithm and based on that decide which will suit best for your data. But in our case, we have already segmented the cells and we have label or mask images. So we can just use directly a label image detector. And now if you do preview, you'll be able to see. So this is the spots. The pink outlines are the spots that Trackmate has detected based on the cellpose labels, which looks quite good. So now it has detected. So the preview was just for the first image. Now it has detected for both the images. And now it's calculating the spot features. Um, so these are some of the filters that you can set. So if you just, yeah. So I'm just removing all the filters I had previously set so I can show you how to do it. So here, if you want to visualize the spots better, you can color it on different features. So the ones with the yellow outlines have more area compared to cells with the blue outline. So this will help you when you are, sorry. So this will help you when you want to set um, thresholds to filter out spots. So in our case, the wrong segmentation that cellpose did were all small in size. So we can set a area filter and say that only spots that are above the number here is 0.79 will be considered as true spots and the others will be removed. So now this was the wrong segmentation that we saw, which is now removed after we have kept this area filter. So other than that, I don't really see any wrong segmentation. So I'm not going to use any more filter. So for this data set, just an area filter has worked well. Okay. So once you have set the filter, just do next. So these are the different tracking methods. This was the um, method that we covered in the lecture where it assigns costs based on different parameters. And then so this is a method where you have different costs. So there are penalties for um, different features. And this was another method that we covered in the lecture where it just sees which two spots are closest in distance between the time points. But this method is the problem or not the problem, the limitation is that if a spot is missed in one time point, for example, there is this, consider that maybe the cell in the edge has moved out of the frame in the second time point, and then it has again come into the field of imaging in the third time point. So then there is a gap in the track. This method does not allow for any gaps in the track. So if this cell has gone out and come back, then the algorithm won't be able to detect that the same cell in the first time point has come back in the third time point. It will start a new track instead. So for this kind of data, if you know that the cells in your object are not going to change their physical features that much, then you can use the overlap tracker. So basically, it measures the area and shape parameters of all the objects, and it will compare between two different images which cells have the closest area, closest perimeter, and so on, and those will be detected as the same cell in subsequent frames. So that's what they explain here. It generates links between spots whose shapes overlap between consecutive frames. When several spots are eligible as a source for a target, the one with the largest IOU is chosen. So since we just have two images, we can use the precise setting here. And now it is done. So you can see that it has worked fairly well, but there is one problem here. So here cellpose has detected two different cells, but in the next time point, it has detected it as a single cell. Um, so we can go back to the segmentation and see if it is one cell or two different cells. So it's this cell here. So it does seem to be one cell. But somehow in the first image, it has detected it as two different cells. Let's see if we can find it here. So these two, here it has detected it as these two cells. Okay. Mm, Let me just open these on Fiji to see what's going on. So this is the advantage of having a visual output. You can check and see if segmentation and tracking have been done correctly. So I think this is the confusing cell here. Yeah, it has segmented it wrongly in the first time point. So one way to fix this is that you can manually add or remove masks on cellpose. So you can go to click select, click on this mask, these two, and then do done. And now this has been removed. But even by looking at it visually, it's a bit confusing here whether it is one cell or two cells, because in the first time point, you do see a different compartment here. So it is a bit confusing, even to the naked eye. So it might be just better to remove the segmentation in this area from both the time points. So I'll now update the masks with the manual correction. And here also we can remove the big mask. So I had not saved the .numpy files. That's why I have to do the segmentation again. But if I had saved the .numpy or .npy file, then the outputs would have already been saved. Yeah, so this is the confusing cell. Again, I'm going to do region select. Sorry, click select. Select this mask and delete it. And now save the updated masks. Yeah, so now both of these masks are updated and we can use this for Trackmate instead. Use this. Now same thing, make it as a stack and then set the parameters. These were 10 minutes apart. Okay. Now you can go to plugins and Trackmate. Just check these calibration settings, and then you can go next. Same thing, label image detector. Now it's finding the spot features, and similarly we have set an area filter so that this wrong segmentation is removed. And we are doing the overlap tracker because the cell shape properties are not changing much in 10 minutes. Yeah, so now the confusing cell is not there. Now it does look like it has more or less detected everything properly. So it has even detected the dividing cell. So here, this was one cell in the first time point, but it has divided and become two cells in the second time point. So the most of the tracking looks good. I am not able to find any errors. So I'm not going to set any filters on the cell tracks. So now, this part of Trackmate will help you to change the display settings. If you, for example, now the cells are all having a uniform outline color. If you want, you can change it to color it based on the cell area. And if you press auto, the scale bar will be calibrated. If you want, so right now it's just the cell outlines that are colored. If you go to edit settings here and select draw spots fill, then the entire color cell is colored. The cells in blue are cells with less area and the cells in red are cells with more area. But when you do this, the tracks are not that visible. So you can make it more transparent. Now you see that the tracks are visible more. Similarly, you can also color the tracks if you want to see which cells are moving fast, which cells are moving slow. Then you can color them based on their speed. but the lines are still a bit difficult to see. So you can make them thicker. Sorry. Yeah. So these are the cells that are moving fast, which have red lines, and the cells which have blue lines are cells that are moving slowly. And if you want to save the entire analysis up until this point, you have to press the save button here, and it will get saved as an XML output, which will have all the analysis parameters that were used. If you want a CSV file with the properties of the cells at each time point, you can go to spots. So cells with the same track ID are the same cell but at different time points. So yeah, this is the property of the cell at the time is equal to 0 minute. So it was at x is equal to 265 and x is equal to 31. But at time is equal to 10 minutes, it has moved from 265.726 to 265.064 and 31.7 to 32.2 microns. And similarly, then you have the second cell and the third cell and so on. Actually, I wanted to increase the. So if you see when once you select the cell, it also highlights which is the selected cell, but here it's a bit difficult to see. So maybe we can change the selection color to white. So now this is the cell with ID 4097 right here. And this is the cell with ID 4965. This is the cell with ID 4105. So this was how it was in the first time point, and this is where it is in the second time point. So now, yeah, so for each cell, it gives its properties like its x and y coordinates, the radius of the object, mean intensity, um, signal to noise ratio, um, ellipse elongation. So it also fits the each object to an ellipse and finds the ellipse long axis, length of the ellipse minor axis. And then you also get the area, perimeter, circularity, shape index, and so on. So from this data, then you can export it to a CSV and do a lot more analysis. So now we have saved the two main outputs, the XML file and the CSV file. Now you can also save this visual output as a movie. So just go to next and select from here, capture overlay, execute. And if you want, you can hide the image so that the masks are not visible. So it has not exported properly. Sometimes this happens that it doesn't export properly. So just try again. Yeah. Mm. So sometimes it doesn't work when there is something in the background. So I'm just moving everything. it works on screen recording, so it's just make sure that not too many things are open in the desktop. I'll close this and try again. Okay. Yeah, this is better, but still there is this brightness difference. Sometimes it works well for me, sometimes it doesn't. Let's try with white background. Yeah, again, it's making a mistake in these. So this is one problem that sometimes I sometimes encounter with Trackmate. Sometimes it misses a few cells. Um, yeah, let's just. This is worse. Let's try now. Yeah, it's not working at the moment. So if this happens, you can just save the XML file, import it again, and try to execute this. So this you can then save as a movie. Which you can then use it for your presentation and so on. So these are the important steps of Trackmate. I'll also show you more about how to use the Trackmate outputs and so on. It's been a while since the class began, so you can take a short um, 10-minute break, and we'll be back by 11:55 a.m. and resume the class. Okay, let's resume now. So far we have seen how to use Trackmate. So once you have the Trackmate outputs, there are also other um, how you can also do other analysis with the Trackmate outputs. So there are specific um, libraries that you can use. So one is called celltrackr, and I think there's one called Trackmater, and there is one which is based on a Google Colab notebook as well. Let me just find its name. Yeah, it's called Celltrax Collab. So these are some codes that other people have written, and they have kindly made it open source. So you can also use them for your analysis if you want to do advanced analysis like plotting the mean square displacement and then get the diffusion coefficient to see if the particles are the kind of motion that the particles are undergoing. Is it subdiffusive? Is it super diffusive? And so on. Um, Let me just share my whole screen so I can show you some of these codes. Yeah, so the first one is CellTrack R. It's a package in R. So in this course, I'm not going into coding that much because it's more of a beginner's course. But those of you who are interested in further processing of your TrackMate outputs, you can use some of these softwares. And all of them have dedicated GitHub pages that explain how to use them. Looks like my internet is slow. So um, apart from that, you also have TrackMate R. So this will give you things like, so again, this has a GitHub page. So first you'll have to install this package, and then here you have to give the XML output. And finally, it can just draw the cell plots. It can give a data set with multiple graphs. And if you read about it, you're also able to see how it does these. So in this option itself, you have um, an option to filter out tracks and so on. And so this was about TrackMate R, and this is another R package called celltrackr, which also can do it can plot the tracks, it can filter the tracks, and then similarly, this can also plot the mean square displacement and autocorrelation. So this also has tutorials where they explain how to do all of this analysis using their package. Um, and they also have a cheat sheet. No, it's not really opening. So, and if you want a easier version of this without codes, you can use this. So this is the GUI version of it. So where you can inspect the tracks, quantify, plot different heat maps, directionality, etc. So here, if you just choose which plot type you want, it will do it. Maybe I'll try opening it on a different browser. I think this would probably be the easiest method to analyze cell tracking data because this does not involve any codes. Let me see if I don't think I have Firefox. Yeah, I think it's not opening so well on Google Chrome. Mm, where was cell tracker? Yeah, I need the cell tracker. Okay, so they recommend Firefox or Google Chrome, but even Google Chrome is not. So let's try importing the tracks here. So I'm not. I think maybe I'm not able to upload because of some settings here. Okay, maybe in the next class I'll try to figure out how to use this. Somehow I'm not able to use it on this particular browser. Um, Let me try using it in a different browser. Um, because that browser is affiliated to my institute. What is this? Yeah. Let's see if this works here in this browser. It's still not secure here. Okay, so they have the options here. Okay, for this we will need the data in this format. Yeah. So to get the data in that kind of a format, you can use this option, export to motility lab spreadsheet, execute, and just select copy everything, and let me try pasting here. No. Okay. So I've just pasted everything here, and now it's in our case, the particles are in 2D, so it has just plotted the particles. So you can see that there are two. Um, so this is the particle at time 1, and this is the particle at time 2. And similarly, the particle meaning, so here it has plotted each of the cell just as a dot of the centroid. That's why it looks much closer um, and much smaller compared to the original data set we have. So now I'm just saving it here. So now that the data is saved, I'm choosing this for the analysis. So somehow this view track display is not working very well. And So let's try plotting something. Maybe the mean square displacement. Somehow it's not really displaying anything here. Yeah, it hasn't plotted anything. I think the data that I uploaded is probably still changing. Drag and drop file names. I don't want to delete this file. What does this do? So if the data had saved completely, we should be able to see it, but it's still, we are not able to see it. So maybe I think we have to wait for a while for the data to properly upload, and then hopefully we'll be able to use it. So, and here in this tutorial, they also have how to use this data, how to write a code to do their analysis. And if you go to their cheat sheet, it has a summary of all the analysis that can be done using cell tracker. And this is a summary analysis of all the analysis that can be done using Trackmate, which is also a R-based package. And for this is the function. For each function, the GitHub page itself has definitions of what should be the input, what are the outputs, and so on. And you can also use it if you have multiple cell tracking files, then you can also cluster them into folders of this kind, and you'll be able to compare the data sets. Um, I'm just seeing if it has if they have the complete source code somewhere. So then we can also see exactly how they are generating all of the MSD plots and so on. But I'm not able to find it here. Okay. So let me try this again. Use this. Mm, yeah. Yeah, I think the data has still not saved properly yet. So um, let's try a different analysis. Maybe heat map, Trackmate. Yeah, it's not really giving any outputs at the moment. So this also has data from other papers. So let's try if this works. And let's try to analyze the data that we imported here. Yeah, so this is working fine. Um, so this is how this first data set looks like, and the second data set looks like this. So ideally, we should be also able to see the data set that we uploaded. I think in our case, we have more than 400 particles. So it's taking time to load everything. So let's just wait for this. Meanwhile, the other software which you can use to, okay, this is working. Analyze cellpose, Trackmate outputs is called CellTracks Collab. It's a Google Colab notebook. Um, just open it. How do I? Mm, yeah. And they also have tutorials on how to use it with Trackmate. So for all of these codes, the input that you will need is an XML file. So they are asking for the Trackmate XML files here. So one option to use this code is to use the input the XML file, or you can also input the CSV file. And then they have, so you can also analyze multiple tracks, multiple tracking data with this. What is this? Okay. So they already have the code here. You just have to run it. So this will also be quite easy to use because they have already written the code. And if you want more information, you can check out the GitHub page to see what kind of analysis can be done using CellTracks Collab. So they also have a video with if you want. So they're showing how to use their code and what are the main analysis that can be done. So it can draw tracks. Actually, I think it's all in the notebook directly. So maybe I'll also add this here. So for those of you who are interested in analyzing the tracking data more, you can also check out this link. And basically, you just have to run this code. So these are some of the metrics that you can calculate for each track. And then you can calculate the directionality, tortuosity, or how straight or how curved the path is. And you can calculate the turning angle of the paths, the spatial coverage, or how much space a particle covers when it moves, forward movement index. Yeah, but if you want to plot MSD, then I don't think this package would work. But if you want to calculate how the cell tracks are the cells moving straight, are they moving in curved paths, etc., and so on, you can use this software. But if you want MSD, then CellTrackR or TrackMate would be better. So still not. Yeah, this has still not worked. Yeah, I think it might be because we have too many particles here. We have almost 450 particles. So maybe I'll try just using the first 10 particles, if that works. Manage files, import tracks. Tracking ID, time point, time, X position. So the format is the same here. That's why we exported it in this format, specifically for Motility Lab. Okay. Save this. Mm, yeah, this is also not working. I'll just wait for a while to see if this comes, if this needs more time. But yeah, these are some of the other steps that you can do after doing tracking. Um, so I have some codes for Trackmate R. Let me see if it's on my laptop. I can close this. Yeah, so this is something that I had already done. So so the only input you need, so if you already have the code, it's easy to run, but to run the code, to write the code, you'll have to know a bit about the software. The only thing that you need here is in this code is the XML output file. Uh, okay. And once it generates this output, it will show up here. But this analysis takes a while. So let's wait. So this is using Trackmate. So you can see that the code is still running over here. Mm, and for this cell tracks on Google Colab, you will have to give the um, folder path, and you can choose this file format, and finally where you want the outputs to be saved. So this is if you want to run it online on Google Colab. I think they also have an option to run it as a Jupyter notebook offline. Um, yeah, cell tracks Colab. Yeah, this is if you want to run it offline without internet, without Google Colab. Ah, okay, this is a video. So depending on your preference, whether you want to run it online or run it offline, you can use whichever you want to. Mm, so this R code is still running because this is a huge data set, but it's okay. We can go back to the lecture meanwhile. So we had used, so now let's try using different tracking algorithms to see how they do. Um, yeah, and set the properties. Uh, I'll just save this so that next time uh, easy to open. So it's saved here. Hmm, what is before? Sorry, guys. I'm not sure why it's saved as RGB. Um, okay. Hopefully, I'll just save this again. Now let's try tracking it with a different algorithm. So the method worked well here, um, because the cells don't change shape much between these two time points. So, sorry. So I'm sorry. I have to upload the cellpose output images, not the raw images. Uh, yeah. Okay. I will save this. Mm, Replace it. Now track it. So one thing that's very obvious from this data set is that um, the nearest neighbor algorithm may not work so well for this because the particles themselves are very close together. So that algorithm may just mistake neighboring cells as tracked cells. So also, so this mask detector does, because the cellpose output here is a label image where each cell gets a distinct number, but this mask detector can only distinguish between mask images of 0 and 1, or 0 for background and 1 for the objects. So if you want, we can try this, but I don't think it's going to work so well. So right now this is slow because the code is also running. Um, yeah, this is, you can see that it's still running here. Everything is slow. Let me close bunch of stuff. Now it's stuck. Wait. Full script. Word can be closed. I'm just closing everything so that there's more memory for everything to run. Full squid. Okay, nothing is responding now. I hope you all can still see my slides, right? Yeah. We'll just have to wait a bit. Everything has become slow now. But probably those are, because that is probably making everything slow. Yes, I want to quit. Mm, all right. We also quit this. Mm. Okay. Mm, yeah. Mm, yeah. So when we use a different detector, it's not able to detect everything properly. So it has detected everything together as whatever is connected together as one big object, which is not correct. So when you use cellpose labels, you have to use the label detector, then it will do fine. I'm saving it up to this step. Um, yeah, it is saved here. So just before the tracking step, so that we can try different algorithms. So now let's try the nearest neighbor tracker and see how it does. So the cell size here is about 10 to 20 microns, and some of the cells move by more than one cell diameter. Um, so just to be sure, we can give a cutoff linking distance of double the cell diameter so that it also detects cells which move fast. Okay. Yeah, so this has not done too bad actually. Um, yeah, because the cells have not moved much. These are images just 10 minutes apart. So for this cell, it seems to have made a mistake. So this cell has moved from here a little bit to the top. Let me highlight it. Yeah, so it has moved up in the next frame. But because we have used the algorithm nearest neighbor, the algorithm has detected that this cell has become its neighboring cell in the next frame, which is not correct. So, and similarly, it has made a mistake here as well. This cell has moved to the top, but because of the algorithm that we used, it has detected its neighboring cell as the same cell in the next frame. So, this nearest neighbor method, it works well for cells or for any objects that don't move too much. But in this context, it's true that these objects have not moved a lot. But unfortunately, here the objects are too close together, so it's detecting the neighbors as tracked cells by mistake. So it's not a very good choice in this context. So similarly for your data set, you'll have to try different algorithms, tracking algorithms, and see which one works best. Um, let's try the simple lab tracker. Yeah, so you can see that in this method, it has detected that the cell has moved to the top correctly. So even this method has not done so badly. So now I'll try running the R code to give you a flavor of what else. My laptop is slow actually. I can start running the R code and we can wait for, I'll close everything else. And we can wait for 5 to 10 minutes to let the code run. Maybe I'll use this instead because this just has 2 time points. But this itself would be quite heavy because there are too many, like there are 500 particles in this data set, 490 something, almost 500 basically. So the code, this has to be within quotes. Ah, okay. Yeah. So let's just wait for some time to see the output that we get here. Okay. I don't know if you're able to still see my screen. Okay, you're able to see the screen. Great. Yeah. So the recap quiz is on mostly on cellpose and some questions on some earlier lectures. So most of these are questions are similar to what we covered in the last week's recap quiz. First question is, which of the following statements about cellpose are correct? Cellpose predicts vector fields, which is true. That those are the flow fields that you get. And cellpose requires manually annotated training data for every new data set. That's not true. There are already models that come with cellpose, which works for different types of data sets. Diameter strongly influences segmentation quality. This is also true. Cellpose outputs probability maps and flow fields. This is also true. And cellpose performs purely intensity threshold based segmentation. This is not true. There are other intensity based segmentations like Otsu thresholding and so on, but cellpose is based on deep learning. So option E is not correct. The second question, when performing 3D segmentation, which statements are true? Images must be provided as a Z stack. That's true. And then we also discussed the anisotropy parameter, which is the ratio of the scaling in the Z, sorry, Z direction and XY direction. So option segmentation is more accurate than 2D slice by slice segmentation. It depends on the context and the kind of image you have, but here it says always, so it's not a very correct statement. Memory requirements increase significantly in 3D mode. Which statements are true? Yeah. So this flow fields being computed independently or not, that depends on whether we set do underscore 3D, the parameter, to true or false. So it's not always independent. It depends on what setting we choose. But the default setting is that it calculates it independently. And how do you choose which cell pose model to use? So that will depend on the cell morphology and whether the objects touch or overlap. The other parameters don't really matter much. So the diameter is an important parameter which leads to either over or under segmentation. Also the flow and cell prob. So these are some of the parameters that you can tune in cellpose itself, and also which channel you use to segment. So if you use a channel like a tight junction protein or any junctional protein, then the cell borders are more clearly visible and the segmentation will be better. So rather than if you use a cytoplasmic protein as the channel to segment, then it will probably lead to under segmentation. So this question is about the difficulties of cell pose. So this is true. It doesn't work well for elongated cells like maybe neural cells or bacterial cells. And yeah, it is sensitive to background noise, and it depends on the quality of the data. And it does have touching cells. Some of the examples that we were segmenting and tracking today were tracking cells. And this question is about scale bars. Yeah, this is true. If you just put a scale bar as 50 pixels, that doesn't make any sense. So you have to calibrate it for the scale bar to make sense. Sorry, this is true. Yeah, and it's better to add it after you change the image size and everything because it does not resize automatically. and it can be burned into the image if you flatten it or draw on top of it. Burned into the image meaning that the intensity values change wherever you add the scale bar. Option E, scale bars should never be added to videos. That's not true. You can and you have to put scale bars to video, so this is not true. So I guess the rest of the questions are not so difficult. So option, this is about lookup tables. Option A is not correct. It just changes the visualization of the image, but the pixel values don't change, so it's not correct. Option B is true because rainbow LUTs or any multicolor LUT will have different colors, and it will be difficult for somebody who sees an image to interpret uh, what it means, where the intensity is high, where the intensities are low, and so on. Yeah, and you have to use the same LUT, so that it's easy to compare. And yeah, you have to write about which LUT you have used in the figure legend. Required for segmentation, not really. It's the absolute pixel values that matter more. And this is about time lapse. Yeah. So if the images are if the pictures are taken every 10 minutes, then the timestamp should also be for every 10 minutes. You cannot change it. This is not true. This is for adding the scale bar, not for time lapse. This is true. So if you, instead of 10 minutes, uh, you give a timestamp of 30 minutes, then people will think that things are actually happening slower than the reality. This is also true. Timestamp can be added as overlay or burnt in. Again, you have to flatten it to make the timestamp as to burn in the timestamp. And timestamps are not optional. So whenever you put videos on your presentation or in your paper, just make sure that it has a timestamp on it. So we are almost at the end of today's class. Maybe we can take up the rest of the questions in the last class, which will be on next week, Saturday. If you have any questions now, we can take it up. Otherwise, we can finish the session for today. Okay, there are no questions, then we can finish the session for today. And the next session is on next Saturday. Thank you. Hello.